Hi everyone, my name is Raikov. I'm a postdoc at the Schultes lab at Stanford University, and I'll talk about our computational project of modeling the hippocampus and information processing in the hippocampus. The hippocampus is a region in the brain that is responsible for spatial navigation and for the formation of episodic memories. Episodic memory associates context with location, and it allows us to remember where particular events occurred. And we are trying to understand the hippocampus in terms of the intrinsic properties of its neurons, as well as the input-output transformations that it performs. The hippocampus receives two primary input streams, which have been called the what and where inputs. It receives them from another region in the brain called the entorhinal cortex. So the where inputs is primarily spatial information, which is active when the animal is running or moving, and the what is context information about objects or other non-spatial cues. The what input is active when the animal is actively exploring objects. And it is believed that the hippocampus takes these divergent kinds of inputs and integrates them to create associations between them. The primary output type of the hippocampus are the so-called place cells, which are neurons that are sensitive to particular locations. And it is also believed that the hippocampus creates more precise spatial representations than the inputs that it receives. So an example of a place cell is during an animal running on a linear track, there are neurons that respond, they fire action potentials only on particular locations on the track, as shown by this firing rate map. And what's also really interesting is that if you look at the entire population of place cells, they all have a preferred location somewhere on the track. And looking at the population activity allows us to decode the position. So there must be some mechanisms of coordination that allow the entire trajectory to be represented and to be associated with various sensory events. Okay, so our lab has a Brain Initiative grant that's focused on a specific aspect of hippocampal information processing. However, that is a process that is very fundamental to the formation of episodic memory. So what happens is that during active exploration or running, the neurons in the hippocampus activate as the location to which they're sensitive is reached. And at the same time, there are very strong oscillations in the electrical field potential of the hippocampus. And it turns out that every place cell is actually active also on a particular phase during these oscillations. So it is believed that this mechanism is a manifestation of this sequence generation that allows us to have the trajectory represented as a sequence of place cell activations. But what happens when the animal pauses to rest or to plan its next move is that there are these very high frequency oscillations that occur. And during those oscillations, the same neurons that are active on the track, on the trajectory, get replayed at rapid pace again. So it is believed that this is a manifestation of the process that forms the representation of the trajectory as a sequence of activation of neurons. And it is the first step through which the hippocampus transfers this information to the cortical, to its downstream cortical areas, which starts then the process of consolidation, that eventually there is a representation that associates events with that trajectory. So it is thought that all of these activations in different environments and so on get transferred to the cortex, to the neocortex, and eventually get consolidated. And disrupting these events also means that no episodic memories are formed. So we are focused on the hippocampal representation of these events, but the intrinsic ability for generating sequences is thought to be very fundamental to the cortex as well. So we hope that this could also generalize to other brain regions. Now, I should have pointed out that in terms of experiments, experimental neuroscientists can study the behavior of individual neurons and maybe record from a few hundred neurons at the same time. But when it comes to studying the interactions between different brain regions, the current experimental neuroscience cannot really cannot make large-scale recordings. So to evaluate the hypothesis of a memory formation, for example, it's really only that it can be done through computational models, because the experimental scientists don't have the technology to record from thousands or millions of neurons. So we basically constrain our computational models with experimental data, but ultimately the evaluation of a theory happens through a full-scale computational model. And so some of the founders of the field of computational neuroscience have postulated that understanding neural system really has to happen on several different levels that are complementary and interdependent. What they call the computational level is the representation of the task on a cognitive high level. What is the animal doing? What logic is used to solve it? On the algorithmic level is usually is understood the representation of information across populations of neurons and how they interact with each other. And finally, what is called the implementational level is what are the properties of the neuronal circuitry that actually implements these algorithms. So we have been using this kind of structure in informing the construction of our computational models. We have a number of sub-projects in constructing a full-scale computational model of the hippocampus. On one hand is the creating a detailed computational representation of the anatomy, which also determines the connectivity and the structure of neurons in the model. There is also, we're investigating the hippocampal spatial and temporal coding on different scales. Every class of neurons in the hippocampus has particular information coding properties, and how they integrate together to create the overall representation is still an open research problem. And of course, it's critical to understand the biophysical properties of individual neurons and the synaptic mechanisms that connect neurons and create microcircuits. So we have also created a multi-objective optimization framework that we hope to constrain these models and make them more biophysically accurate. It is necessary to do this kind of multi-objective optimizations because the typical neuron models have hundreds of parameters, which requires tens of objective functions. So on our computational geometry project, we had previously created a parametric surface representation of the anatomy of the hippocampus. The hippocampus has this sort of a curved tube shape, which with varying thickness, which also puts particular constraints on the number of cells. It also creates certain constraints on the shape, on the morphology of the neurons. So here we generate 1 million unique morphologies for each cell in a region of the hippocampus. These shapes, they have particular implications about information processing. And furthermore, the connectivity of the neurons is determined by these experimentally determined distributions of axons, which are also dependent on the geometry. So all of these components have some implications about information processing. And we are the first lab to really start addressing them at this level of detail. And then furthermore, the actual neuronal properties and circuit properties are of course critical in determining the information processing abilities. This is one of the circuits of one of our models of the dentate gyrus, which has at least 10 different neuron types, which they mutually inhibit or excite each other given different kinds of inputs. Which creates very complex nonlinear interactions that essentially keep the network activity sparse, and essentially they ensure that there's always salient signal. This particular network is of size 1.2 million neurons, about 18 billion synapses. There's at least 4 equations per synapse, so very quickly these become very large models that can only be simulated on a machine like Blue Waters. And then we also, of course, want to understand the hippocampus in terms of the input-output transformation. So we currently create representation of the inputs that come to the hippocampus from the entorhinal cortex. These are cells that are called grid cells. They're spatially sensitive at different locations, and these locations are on a hexagonal grid. These provide sort of a very overlapping and noisy spatial inputs to the hippocampus, and the hippocampus then resolves them and sort of cleans up the signal. So you can see one representation of this trajectory. These are the activations of all the inputs to the hippocampus, and they're sorted by the peak firing rate. And you can see that there are also multiple representation of this trajectory. So this noisy input then gets transformed into something from where the trajectory can be decoded. So when given this input into our full-scale simulation of the dentate gyrus, the region of the hippocampus on blue waters, the principal neuron type outputs, these are also the outputs of the principal neurons in the model, also sorted by the peak firing rate. So they represent the trajectory a little bit better. This is a histogram of the spatial sensitivity to each cell in the model. And the vast majority of cells are actually not, they don't respond to particular locations. And then the largest number of cells that are sensitive, they have one place field. They're only sensitive to one location. And this has been consistent with experimental results published just last year. So from that aspect, the model is starting to get to be reasonably constrained, although there are still some other constraints that we're working on resolving. And furthermore, the activity of the network is rhythmic. This is of importance to neuroscientists because it is thought that this rhythmicity allows cells to have a particular phase preference from which that means less interference in the network, essentially. So these are biophysically plausible, at least, properties of the model. And this is really the first time that a full-scale model of the rat hippocampus has actually generated some interesting information processing. Most previous models have either been reduced scale with just a few hundred neurons, which leads to distortion in the dynamical properties of the network. So this is really the first time that we can see something realistic is happening. But we still need to work on ensuring that our cell models have biophysically accurate behavior in a variety of cases. So we have also developed a what we call nested multi-objective optimization, which is a form of Bayesian hopping. We use essentially evolutionary optimization with simulated annealing to sort of fine-tune the local optima found. And this has now produced, so originally using the Bayesian hopping optimization strategy from SciPy, we needed on the order of 1,000 iterations to start converging. But with our framework now, it's on the order of 200 iterations. And so these are some of the constraints that we fit the models to. The response of the neuron to different frequencies of stimulus are very important and have implications for information processing. So there have been a lot of experimental results published recently that allow us to constrain the models and ensure realistic biophysical behavior. But these optimizations are still fairly computationally expensive, so we are still working on trying to optimize them further and speed them up. But we've already published on GitHub our optimization framework, and hopefully there will be a publication as well. Another very important aspect, both for computational efficiency and information processing, is the synaptic mechanisms. So there are processes called facilitation and depression that essentially where the synapses regulate, if there are a lot of inputs coming to the neuron, the synapses regulate the conductances so that there's no sort of exponential blow-up of activity in the network. In computational neuroscience, the details of these processes have often been ignored, and people just use simplified models. because these are also computationally expensive. They require keeping track of past history, a lot of state variables, which when you have 18 billion synapses, you cannot really do this. But we devised analytical approximation that allows us to keep track of the activity without this blow up of state variables. And now, even though Blue Waters could simulate realistic synapses where we had one set of equations per connection, it was taking maybe on the order of 24 hours for very short simulations. But now with our new synapse mechanisms, we have for 5 seconds of physical time, we need 3 hours of simulation time on Blue Waters on 2048 nodes. So that's maybe a 3 to 4-fold improvement in performance. And then finally, another very important aspect is the distribution of synaptic weights, so this is one aspect where experimental neuroscientists have not been able to measure, what is the synaptic conductance of every neuron in a particular brain region, because there's no technology to measure this. But there have been small data sets that tell us something about the distributions of these synaptic weights. And they are usually not Gaussian, they're log normal or similar. And so our computational modeling infrastructure allows us to generate hypotheses about the structure of the synapses in the entire network and investigate what are the implications for information processing. And nobody has really been able to do this before. We've only now established our infrastructure to be able to investigate these questions. And so it will be interesting to see how these global shapes of the weights between neurons are going to affect the dynamic behavior and information processing properties of the entire network. And finally, we have been developing our framework for parallel I/O and analysis and creation of neuronal networks. We started, thanks to the Blue Waters paid program, we developed an initial prototype last year. And this all worked out so well that we've been expanding our infrastructure. So now we have a lot more support for 3D volumes and geometry, various kinds of synaptic generation mechanisms. Our optimization library is also now based on this parallel HDF5 for saving parameters and evaluating models and saving their outputs. And we've also started using it for visualization and plotting. So thanks to the paid program, we've actually expanded our infrastructure and have improved the performance tremendously. And that's all I have. This is our lab. This is my PI, Ivan Šoltes, and thanks to Blue Waters and NCSA, and our funders are the NIH through this Brain Initiative grant. And that's all.